this morning, Dr. Hillengass gave us a little teaser, well, in the Q&A, about spatial heterogeneity. So I'd like you to see if we can all say that. And I have really been intrigued by reading about his research in this area, and really what it is pinpointing myeloma in the body, and asked him if he would teach us about this today. So I'm excited to learn more.
I'm happy that I had the talk this morning before Dr. Shah, because it's always challenging to be after her to keep up the quality. So I hope that I don't drop too much. This is actually something that we're interested in too. I told you that I think about imaging a lot and that we have done some imaging research. And as you know, myeloma is not involving the bone marrow heterogeneously as I would have expected as a young resident. I thought it's a hematological disease, the bone marrow is everywhere in the bones, so the disease should also be everywhere, but it's not, right? So we have some areas where it's not like that. And just waiting for this to respond. What I would like to talk about is why are cancer cells heterogeneous? Because we think that cancer develops out of one cell. Right? Something goes wrong, this cell goes rogue and proliferates and kind of divides, and then you have all the same bad cells everywhere. That's in solid tumors, that's metastases. In hematologic diseases, it's just kind of the name of the game. And why are monoclonal gammopathies like multiple myeloma heterogeneous? Shouldn't they be all the same, as I just said? Another question is, where does the spatial heterogeneity come from? And I will explain why I talk about that. Myeloma is not not the same myeloma everywhere. Dr. Landgren, a mentor of both Dr. Shah and myself, said it's multiple myeloma, right? It's not one myeloma, it's several myelomas. And why does it matter? There's a really nice summary or review paper about this topic of cancer heterogeneity. And it is really relevant because I think it's one important factor why we cannot cure certain cancers. And that's kind of our approach to deal with that and try to figure that out. These are hypotheses. There is some underlying science supporting that, but I think it's a very good illustration what might happen and what most likely happens in certain cancers. On the top panel, you see initially there's kind of a normal tissue. Then there's something going wrong, like radiation exposure, toxic exposure, whatever triggers that. And then certain cells, something goes wrong, and they kind of develop into cancerous cells. And then they take over, right? They grow and replace the good ones. And that's exactly what's happening in myeloma, at least in part. So the myeloma cells take over, and the healthy plasma cells are kind of underrepresented. And then they progress further, and then we have treatment, and that triggers further mutations. Another thought is that there's this hit again, radiation, whatever, toxic, and then certain cells go rogue, but then those cells mutate further and develop into a further kind of spread out more and become more diverse, and another subset of these cells also spreads out. That's also leading to heterogeneity. So either one set of cells just take over, or one set of cells start to proliferate, and then they already become more heterogeneous. Because cancer cells, usually our cells have intrinsic mechanisms to check if everything went well, right? When they divide, they have to divide their DNA, the plan, the blueprint of the cell. And if something goes wrong there, usually the immune system recognizes that, certain other mechanisms recognizes that, and then they repair it. And that's what doesn't really work in cancer sometimes. To make it a bit more complex, when we treat cancer, what happens then, right? So we have these cancer cells, the red ones, we treat, and there might be, and that was the question earlier, there might be something left, right? Even with MRD negative, you have seen there are relapses after MRD negativity or on MRD negativity, still the disease comes back. So there must be a few cells that we cannot detect with our current methods, and they start to proliferate again because they have survived the first line of treatment, right? So oftentimes patients ask me, I had such a great response after my transplant, first line, many, many years, and now I have second line, and the disease came back after 1 year already. And say, yeah, the cells that come back at the relapse, and I don't want to scare it, it's just kind of how we think about it and what we have to do with it, those cells have survived already a very strong induction treatment. And I completely agree with Doug that we should do our best treatment up front, because that's where we have the least mutation, that's where we have the best chance to hit as many of those as possible. Another possibility is, and again, these are hypotheses, we achieve a maximal response, for example, MRD negativity, but there are a few lingering cells. They might have died if we had treated more aggressively, but at one point toxicity kind of limits us and limits all of you, right? So they might linger, they might have responded, but then those cells are still there, and then they progress and they kind of mutate and become different in their setup. So that's another option. Because of our treatment, we might trigger something like that, right? And it is true to a certain degree in myeloma. This is actually a fairly old study where they did a lot of bone marrow biopsies in one poor patient, but we learned a lot from this. What they call this is the clonal tiding of myeloma. So myeloma consists of several clones, and you see in the beginning it's mostly this red clone, right? There are a few other ones. So already at the beginning, they found different clones. They did sequencing and they found different clones of cells, so different kind of types of these myeloma cells. Then they gave a treatment, and you see that this red clone responds, but the orange one doesn't, right? So then they changed treatment because the patient relapsed. And this, again, this is old data, but it's still true, right? And you can see that all of a sudden this green clone comes up, right? And that's just because this green clone is more resistant to the next line of treatment than the red and the orange ones, right? And then so on, and eventually in this patient, this dark blue one takes over, and there was actually a plasma cell leukemia, which is the most aggressive version, and we see that in our practice, that after several lines of therapy, the disease becomes very aggressive, causes soft tissue tumors sometimes. Again, not to scare you, just to know we are aware of that, and we look for those things later on in treatments. Back to this really good review about the heterogeneity. To make it even more complex, it might be that there are different clones in different parts of the body. For example, with metastases in solid tumors, if you do a biopsy of a metastasis of a breast cancer, for example, and compare that to the original breast cancer, that might already look different, right? Because in a different area, we talked a lot about the microenvironment. There's a different nurturing environment in, for example, I don't know, the bone marrow as compared to the breast, as compared to the colon, right? And so where a cancer cell grows also has an influence on how it grows and how it develops, which again makes it more complex. And then there's, again, also the first thing is actually what I showed for myeloma already. Where does myeloma come into the play? As I mentioned, myeloma is not always homogeneous. It's sometimes heterogeneous in the bone. And these are MRIs of the lumbar spine. And you see these blocks. These are the vertebral bodies. And usually what we have, I think almost all of us in the room have a fairly fatty marrow. The younger ones have less fatty marrow, and I hope I didn't offend anyone. The older we get, the more fatty marrow we have in our body. And that's actually what I mentioned earlier when we talked, the two of us, right? And this is usually how it's supposed to look like. But I have patients with myeloma who look like that in MRI. Then I have patients who look like that. You see, it's darker. Darker in MRI means more water. More water means more cells. And in this case, more myeloma cells diffusely infiltrating the bone marrow. And then I have patients like this, where you see it's the brighter background. It's called hyperintense. But then you have these dark spots. And those are, we did biopsies, and I'll show you more about that later on. Those are myeloma cells, right? So accumulating as so-called focal lesions as compared to the diffuse infiltration that other patients show. And to make it even more complicated, sorry about that, you can have the diffuse infiltration and on top of that, the focal lesions, right? And we looked into the literature, again, some time ago, but it's about 20-30% of all patients have this minimal involvement, have the 30% have this diffuse, 30 about the focal, and then we also have some with this mixed. So it's all over the place, basically. You can find everything in every patient. We don't even know why. We compared that with histology because when we do bone marrow biopsy, our pathologists tell us how the bone marrow looks like. They say sheets of plasma cells. They say small nodules of plasma cells. And we compared that. We compared the MRI with, and it's not super close correlation, but we found that there seems to be a biological background because most patients who had these nodules in the bone marrow of plasma cells also had these focal lesions in imaging. Right, so that's something. There seems to be a biological background, and we don't know is it the myeloma cells or is it the microenvironment or both, and that's what we try to figure out. There are different prognoses. I mean, the normal-looking bone marrow seems to be better because it's closer to normal. The nodular or focal lesions seem to be a bit more worse from a prognostic perspective. The diffuse, even worse. It seems to be just more tumor burden. And then there's the mixed one that seems to be the worst. This is, again, very old data, but it shows, again, there seems to be a biological background about that. Why is it relevant when we talk about MRD, when we talk about great response? The problem is if we do a biopsy, a bone marrow biopsy, we usually do it here, right? And everyone still feels the pain. I'm always bragging about that. I will brag here as well. I had 5 bone marrow biopsies myself for research, so I know the pointy part of the needle. I have done over 1,000, I think, on the not pointy part, but I know how it feels. And the problem is we might do a biopsy here and hit one clone, if that's such a focal lesion, you see it's a darker spot, or we do a biopsy here and this is a different clone. Because we build a lot of our treatment based on these assumptions that we say, okay, we do a biopsy at one spot and then we treat accordingly. It might not treat everything, right? So that's why it's important. And also you can replace the name clone 1 with MRD negative and the name clone 2 with MRD positive. If a patient is MRD negative, and that's what Dr. Shah already mentioned, we also have to confirm that they don't have any kind of focal accumulation somewhere else in the body so that we don't miss anything there. So the best is to have MRD negative in the bone marrow, but because it's very sensitive, but also rule out that there are other lesions in the body that we just missed with our needle. Okay. And we have done that. We have done a study, and we're still doing it, biopsies of focal lesions. This looks a little bit barbaric. This is a bone marrow biopsy. We do a bone marrow biopsy and a focal lesion biopsy in patients where we are not sure if this focal lesion is real, if it's maybe a second cancer. Unfortunately, in Buffalo, in the Rust Belt area, we have a lot of patients who have more than one. I have up to 5 cancers in one patient. So sometimes I really have to make sure that I'm only treating myeloma, not something else. So that's what we do there. And I have to admit, my patients, I have not had any complaints about these procedures. Our radiologists are great. You know, the hematologists are kind of like the blind man going in somewhere, like I palpate it there. We do that a lot, so we have kind of a good feeling. But the radiologists actually know where they are going, right? So they are a little bit better, and they use a drill, which sounds horrible, but it's much quicker, and they have better samples. So I have not really had any complaints about that, and so we could learn more about myeloma. And this is how it looks like. You see this tiny spot where the red arrow is, and the radiologists actually hit that spot, and we found plasma cells there more than in the regular bone marrow that we did on the other side. Right, so the focal lesions, and oftentimes fellows ask me, what's in the osteolytic lesion, right? And initially I said, oh, I think myeloma cells, but now we can at least prove we actually found there are myeloma cells in there. Colleagues from Arkansas, now they are back in Germany, 2 Germans, kind of raided the biobank in Arkansas, in Little Rock, and they found already in retrospective analyses, fine needle biopsies, Bart Barlogi, we talked about him, he did biopsies in a lot of areas of a lot of patients, and then they compared and they found there is a genomic heterogeneity. The cells can look different in different areas. This was retrospective, and they looked into 200 patients, and there were only 40 that they could use, so it was a little bit challenging. We said, okay, we do that prospectively. And I studied that in Heidelberg and then brought it to Buffalo. And Maximilian Merz, you will see his name a lot because he has done an amazing job. He's unfortunately back in Germany, but we still collaborate, and I try to bring him back to the US. He has done a great job in analyzing all that stuff, because if you do genomics and if you say microbiome analysis, that's very challenging. You need really, really brainy bioinformatics people to analyze this terabytes of data, and I don't even pretend to understand all they do. But he did a really great job in that. And you can see here on the right side, the dark areas are myeloma cells, and you see in the focal lesions there were many more than in this random bone marrow biopsy that we also did at the same time. This is just showing where we all did the biopsy. Sometimes, like, there was a lesion here and we didn't know, is it really myeloma? Sometimes it was the only lesion that the patient showed at relapse. We did a biopsy to confirm that it's really the myeloma, because sometimes, as you know, we have this non-secretory disease where we cannot measure the M spike or the light chains. So there we needed the biopsy, and that's the area where it was always a clinical indication, and that's where we did the biopsies and always compared with the bone marrow. So that was the analysis. We don't have to go into detail, but we did whole exome sequencing where we looked for certain areas in the whole gene, not whole genome, but the whole exome, that's the expressing parts of the DNA. Then we looked into single cells and looked what was expressed there. And again, it was a huge effort that Max did there. One interesting finding was for me as a bone myeloma doctor, myeloma bone doctor, is that in the osteolytic lesions there were certain genes that are kind of responsible for bone metabolism were overexpressed. Again, we don't know why they were overexpressed, but at least it fits a little bit more in the picture where the bone is destroyed. There, the myeloma cells have these certain genes upregulated that then stimulate the bone turnover. So there seems to be a biological background about that as well. Another interesting thing that fits to what I talked about in the beginning is that when we looked at relapse, it's on the right side, the 3 patients, every bar is 1 patient. You can see the a little bit lighter area is where they have non-shared mutations, so meaning they were not the same in both areas, and the darker part is always the shared ones. So you see at relapse, when the disease comes back, then there are less shared mutations. So this spreading out of kind of different clones developing over time seems to be real and seems to be also happening in multiple myeloma. But we could not explain everything. At first diagnosis, it looked very, very similar. There were certain unshared mutations, so there was a little bit of a diversity, but not that much. So we said, okay, if it's not the myeloma cells, or at least not the myeloma cells alone explaining this heterogeneity and this different growth even, we have to look into the microenvironment. And so we looked into the microenvironment in the sense of the immune system, so certain T cells and other cells that I mentioned earlier that we also want to influence in our exercise study, and they were different. And we saw again after therapy, this was I don't want to say something wrong. Right, this was blue was first diagnosis and yellow was relapsed. And you can see that the blue is oftentimes higher, so some of the controlling immune system cells that are controlling the cancer are sometimes high, or most of the time higher at first diagnosis than they are at relapse. And that's something that we discussed earlier, that the immune system over time with our treatments unfortunately gets exhausted, and we see that in these biopsies. This was not different areas of the body, this was before and after, at first diagnosis and at relapse. And also before and after therapy, we saw similar changes with patients who were, we had controls, healthy controls, we had newly diagnosed myeloma, and then at relapse, these clonality and richness, so the array of T cells attacking the myeloma cells was the lowest in the relapse patients. And patients who were in remission had a better immune system. And that's something else that I don't have a slide for, but that we presented as well. And again, it was actually Maximilian's wife, Almut Merz, both very German names, by the way. They showed that even vaccine titers became better after treatment. When the patients responded, the immune system got better, and they could mount a better immune response for certain bacteria and viruses. Right? So it is very important to get the disease under control, to get the immune system also the ability to recover. And then here is this immunophenotyping. Again, in remission, some cells went up and so the immune system became stronger. Even though those patients had toxic treatment, the immune system became stronger just because we got the myeloma under control. Right? So low myeloma burden also gives the immune system a better chance to recover. So that's something that we also learned in this study. Another reason why it's important to do the biopsies where the myeloma is, there are certain mutations that we can target. I mentioned earlier in our Q&A that there's this certain gene that is kind of related to the BCMA, or that leads to the BCMA expression. It's this TNFRSF17. So that's related, and some patients don't express that anymore, or never expressed it in the first place. So those patients might not respond as well to BCMA-directed treatment, for example. And again, you see at relapse there are certain mutations that are either there or not there anymore, which again speaks to the heterogeneity and to mutation of the disease over time. But we also have some targets that we can kind of address in myeloma. Even with all the drugs that we have, sometimes we run out of options, and then I personally try to find things that are approved and work in other cancers. And we have studied that actually, we have studied that once in Heidelberg. There's, for example, a mutation called BRAF. And this BRAF mutation is very common in melanoma. So not multiple myeloma, but malignant melanoma, which is oftentimes confused, not in this room, but outside of this room. And it is approved for melanoma. It works very well with this mutation. And now we treated a patient who had, it's unfortunately only in about 4% of myeloma patients, it's there, this mutation. So this patient here had the mutation in this where we did the biopsy, in this whitish tumor. And the patient got this BRAF inhibitor that is approved for melanoma, and it worked in multiple myeloma, to confuse everyone, right? So that was really cool, and I have tried that in patients who are in great shape and say, hey, I'm not done here. Then I look for certain mutations that might respond. And we briefly mentioned venetoclax with the 1114, so that's another thing. But then the problem is, if this mutation is only in this tumor and not in the rest of the body, we only treat effectively the tumor, right? So that's something to keep in mind, where this heterogeneity is also important. And then there's also size matters. I don't want to go into details there, but there's a positive correlation of the size of such a focal lesion. So the bigger the focal lesion, they found, the more mutations are there. So another reason to keep the lesions small and to treat effectively, right? In summary, as I mentioned, there's a genomic or instability, actually. Some have more stable, others have more unstable genomics, and that can lead to mutations over the course of the disease. There is a different composition, and the treatment kind of makes that even more diverse because some cells respond. And it's oftentimes not like the treatment works and those cells are all gone, right? As you might know, if you might remember at first diagnosis, oftentimes the first step with the first cycle is great, right? So a reduction of 70%. And then patients are very disappointed when the second cycle is only 30% more reduction, or which would be 100%, sorry. Bad example. But like the steps become smaller, and that's just because the cells that are more resistant don't respond as well to the treatment as the ones initially that we kill off fairly easily. Right? So that's something that we see over time as well. Then spatial heterogeneity, I told you about the biopsies in different areas of the body. The microenvironment in different areas of the body might be different. And then personalized medicine is what we want, because we don't want to target the whole body, we want to target the cancer cell. But then the problem is, if this cancer cell has a certain mutation, another one in another area of the body might not. And then we kind of trigger this heterogeneity in the resistance. And then again, even the size of the lesions might have an influence. So I kept this a little bit shorter because I have not as much knowledge about it. So, but I think we have more time for questions later on. Thank you so much.
That's a lot for my brain to take in as well. But fascinating to me, and just the potential of the personalization and what that can do, and the impact that can have. Thank you so much for explaining that, Dr. Hillengass. And